in the brain, the most major being the cortex, and primarily the hippocampus, which most of us know about. If not, I'll give a brief intro into that. But the hippocampus is where most models of the brain in learning and memory kind of form from, mostly because the hippocampus is critical for learning and memory. Without it, you cannot form new memories for the most part. Because of a patient named HM, when he had his hippocampus ablated, he was unable to form new memories. And because of this, we are really interested in, okay, so what is this brain region doing that would allow it to serve this role in the brain? And so for continual learning, this is an obvious place to try and look and understand, to fully understand how the brain is doing what it does. And to do so, I use animals, because unfortunately people do not like us to poke and prod inside their brain unless they're in a clinical case, in which case we can. But for the most part, we use animals for this to do all sorts of manipulations. And you'll see on the upper right, that would be a rodent brain. That blue part, I don't know if you can see my, oh, you can. This part right here, moving up and down, is the hippocampus inside the rodent brain. You have the dorsal region and the ventral region. And if we blow this dorsal region up to actually see the circuitry within, you'll see that the circuitry is pretty well defined. And then even this is a really high level view of the hippocampal circuitry. And there's distinct subregions, as some people might know if you've studied this at all, going into it. And it gets a bunch of input from two other afferent regions, the entorhinal cortex, which is a cortical region, which projects heavily to this. And for the first part of what I'll talk about, there's three key regions that you'll need to know. The first one is the medial entorhinal cortex, which is the major input to the CA1 region of the hippocampus, which could be considered, say, the output. You'll see it as information flows kind of in this U-shaped pattern through the structure. But the entorhinal cortex projects to the dentate gyrus, CA3, then CA3 projects to CA1. But also there's another projection that goes to CA3. And so at the end of the day, what you see is this kind of dual projection from entorhinal cortex to CA1 to dentate and CA3, and then from CA3 will project to CA1. So CA1 kind of gets this dual input from the entorhinal cortex and CA3. CA3 is interesting for the simple point that when you look at the anatomy of CA3, you see these reciprocal connections. It's basically a recurrent neural network, and because of this, it's been proposed to be kind of this mnemonic functioning of the hippocampus. But the hippocampal literature is vast, that there's a whole bunch of other stuff that we can unpack later on about other ways that this might happen. So the circuit is well defined. And another note, that the Hebbian rules that you see in Hebbian networks and Hopfield networks and these sorts of things was kind of first formulated from the hippocampal long-term plasticity and these rules where you have these functional strengthening of connections to given activity. And also what I'm really interested in, what we do a lot when we record from this region, so like we would take an electrode and we pull it down into the dorsal parts of the hippocampus and or the ventral parts or other brain regions, is that you see the activity is timed. So you get these, as you probably heard, neural oscillations. And particularly, there's a few that we're interested. One, for instance, is the theta rhythm. So this is activity fluctuating at 6 to 12 hertz. But there's others. There's beta we're interested in. There's also high gamma. So there's a high and low gamma. So these are in the 60 hertz range. And each one of these different neural timings and different oscillations can kind of tell us something different about the brain. So for instance, you have high gamma, which has been associated. If you record from CA1, you see high gamma. It's been associated with the entorhinal region. But say you see low gamma in this, it's been associated with CA3 given like lesion studies, and there's a big literature on that as well that we could talk about. But those are biomarkers of saying at any point in time, where is CA1 getting its input? Another thing that you'll see when you start to dive into the literature pretty much anywhere in the brain is the neuromodulators. And what these do, say neuromodulators will come in and they'll innervate different parts of the hippocampus, the projections from the neurons. And these can tune the activities up and down to add another layer of dynamicism to the network. So one of the earlier projects I worked on in this field, trying to study how this whole thing works, was what we called hippocampal mode switching. That being when you have any, let's say, Hebbian learning system, you have learning, which would be the strengthening of the weights, but then you need to get that information back out. That would be recall, which would be the reactivation of that network, for instance. A problem with this, however, is when you recall some task and you reactivate those neurons, that can lead to runaway plasticity, meaning that even when you recall, given that Hebbian rules imply that whenever you activate two neurons, they would strengthen together, when you recall something, that it would lead to a learning style plasticity, even though you're not really learning anything new. And so this would run away over time and lead to synaptic strengthening that you might not really want. And there's a simple example of this, that you're in like this T-maze task. The animal has to run up to the T-maze. It's in the stem. It could look left or it could look right. And if the food reward's on the left, the animal will run, get the food reward, then it goes back to the stem. And on the next trial, you switch. So you put the food reward on the right side. The animal doesn't know this yet, so it would run to where it found the food last. It would find nothing. So then it would go back and realize, okay, so the next trial, I need to go to the other side of the maze and get the food. And so then it would alternate each time once it learned this task. And you can put the animal in the stem part, and you can determine which way it goes, whether or not it's learned that the food has moved yet. I tried to do this with tabular Q learning methods, and you can't really do this with just your standard Q learning. Although if anybody has any input on how you might do this with a reinforcement learning method, I'd be really interested, because that's something I'm thinking about right now, to try and model what we have these animals do. So an early model that I worked on in computational neuroscience was this idea of hippocampal mode switching. And simply, we just modeled a really high-level version of the hippocampus that I talked about just earlier. So you have an interinal input to CA1, and you have a CA3 input to CA1, which mirrors some of the evidence of CA1 having both of these inputs at the same time, kind of like a comparator between the two. And what we're also interested in is this idea that the hippocampus has been known to switch on this blue theta oscillation, so on these sub-millisecond timescales. But it also gets this innervation, this term right here, CR, is for acetylcholine for the CA3 region, and innervations from acetylcholine from the leftmost region. And these happen on much slower timescales, so in terms of seconds. And so what you see is this kind of dual process modulation between the two, the activity of the network, which would be the theta oscillation. So these is how fast these different neurons would oscillate. And then this modulation return from acetylcholine. And so together, what you get when you're looking at the dynamics of encoding and recall, and previous work has showed that possibly this theta oscillation, which has been prevalent in the literature since, might be a biomarker for the different modes of encoding and retrieval, because when one of these so like when interinal cortex is in one part of the theta rhythm, CA3 has been offset 180 degrees. So its theta rhythm is offset, which would mean that there's different dynamics of plasticity, because when you're in a high activity region, you're going to be a lot more plastic than if you're in a low activity region. And so because of this, you can kind of see this dual process of CA3 talks to CA1, then interinal talks to CA1. And both of these regions also have been associated with interinal being direct input from the brain. If you record from it, the types of activity that you see are more in line with the direct input that the animal sees. Whereas CA3, given those recurrent properties, has been thought to be more of a mnemonic function. So it could serve a literal recall function. And so instead of taking the Hebbian net and making just one network for encoding and retrieval, what we think that the hippocampus might be doing is splitting both of these functions. And so you set the new weights from the inputs from CA1 to CA, from interinal cortex to CA1 for this encoding part. And then you set the recall of the information. The output of the hippocampus goes from CA1. So when you want to recall something, you trigger some of these CA3 afferents. to CA1, and that could serve as the recall function. So when you put both together, if you take the cholinergic modulation on this system, you can kind of do the same thing of switching between this, let's just call it the encoding region and call it the recall region, on slower time scales. And this might be good for really novel situations, for example. So the theta rhythm is almost always going when the animal's moving or when the animal's thinking or doing some sort of task. And then when it's stationary, you see a lot less of the theta rhythm. You see more of like ripple oscillations, which I can talk about a bit later. But what we think is that cholinergic modulation and modulation more generally can kind of serve to set the overall system of the network such that you can kind of tune the activity. It's still in this switching between interinal activity and C3 activity, but at the end of the day, it goes through. And this fits some of the evidence that we're seeing. A big paper was from Colgan's lab in 2009, and there's been a few since. There's also been a couple of rebuttals, which has kind of muddied up this literature a bit more. But you can see here those high gammas and low gammas that I talked about earlier. And what was interesting is when you look at the oscillations, you only see, this is a figure from her paper, you only see high gamma and low gamma in certain, you won't see them coupled. So down here is a simulated time series and output from the model. These would be your slow gammas for CA3. This is your fast gamma from interonal cortex. And you don't see them overlapping hardly ever. And that's what these plots are showing. You see them offset, which is kind of exactly the prediction of our model, that you would see this mode switching between the two regions. And what sets this, the differences between these, the offsetting of the different regions could possibly be cholinergic modulators. So it's a lot, and to really do this properly, I would need a lot more time. But if anyway, it kind of gives you a few idea of the key players that we're thinking about. There's neural oscillations, there's the heavy and learning connections between them, and there's neuromodulators that all come together and form some of these dynamic networks that can help the animal learn different tasks. And most importantly is that if you don't have these offsets, the animal can't, or at least the model can't perform the T-maze properly. Because you get this runaway plasticity, which for me it's not necessarily catastrophic forgetting, but it's kind of a different type of interference that I don't think we talk about as much, which is this runaway retrieval interference of the weights, which would prevent the network from learning any new task or any change in environment in this normal Hebbian regime. Another thing that I've been working on quite a bit lately is a new task that I've been working on is Fortin Lab at UCI. The last work was at my old institution at Indiana University. So this is the stuff that I'm kind of thinking a lot about now. And it's quite literally, so continual learning, Vincino had a really good tweet the other day that was continual learning and sequence learning for networks is pretty much the same thing, right? It's two sides of the same coin. You want a network to learn sequential information is basically the same idea as you want the network to learn over a period of time, say time series data over the lifetime of the network. And so what I'm really interested in is seeing how does an animal do this, and how does the brain, and particularly the hippocampus, do these sorts of tasks. And so we give an animal this sequence task where animals are really good at, particularly rodents are really good at odors. So we make them learn an odor sequence. So we tell them A, B, C, D, but these could be lilac, mint, pick your favorite odor. We have all sorts of different ones that we use in here. And they have to learn this sequence of odors. We know that they learn this sequence because every now and then we'll switch it. So instead of A, B, C, D, we'll do A, B, C, E. And if they detect that this sequence is incorrect, then they'll pull their nose out. And at the end of every trial they get correct, then we give them a reward to encourage them to continue to work. And here's some of the neural data that we recorded from this. It's CA1 of the hippocampus, which I just talked about earlier, pretty common brain region for these things. And so you can record all of this neural information. Here's the theta time series that I kind of spoke about earlier for those who aren't as apt at it. And then we have this neural information. Critically, this is interesting to me because most of the hippocampal literature has been kind of, especially people who I'll talk about later, has been interested in spatial learning, right? So you have an animal solve a maze, a water maze, a Morris maze, or a T maze is still spatial. Place cells have been a key part. But what is just now kind of getting more emphasis in the hippocampal literature, particularly in rodents, not necessarily in humans, is non-spatial sequences and odor learning and odor representations more specifically. And this is because it's really hard, particularly in the part of the hippocampus we're recording. It's harder to determine the coding of each of these odors. So this corresponds to odor A, B, C, D. Can we show that these neurons and this neuronal assembly that we're recording is representing this particular odor at any given time? And with traditional methods, so say the same methods that we use for place cells, you can't really do this, and you can't show that there's an exact non-spatial representation, or at least a one-to-one mapping between the brain state or the assembly recording and the environment at a given time, like you could with spatial information. So what they did, and a lot of this work was a collaboration with our computer science and statistics department to kind of build this thing, was a convolutional neural network, which decodes that information, the neural signals that I showed you earlier at every single odor, and to try and output the probability that the animal is thinking about one of the odors at a given time. And to do so, it worked surprisingly really well. And this was kind of exciting that we could maybe start to look at different brain representations that we haven't been able to do so before. Again, leveraging AI technology to neuroscience, which completes what DeepMind calls a virtuous circle, what I call the AI neuroscience feedback loop, where neuroscience, like we're showing today, can help AI, but at the same time, AI can come back and help neuroscience also. And so what you see here going down is each trial that the animal has. So it's presented a different odor on each one of these trials. And then here are the odors, A, B, C, D. Darker squares represent a higher probability of decoding. And then what we looked at here is from the point of entry to when the animal pulls out. So remember I said the animal puts its nose in a port, and then it smells the odor. So the odor information goes up through the port. And so we can see how the neural representation of each odor evolves over the course of that time that the animal is smelling the odor. And if you take all of these at each time bin and you compress them down into individual probabilities for each odor, you can see this plot here at the bottom, which each line represents a different odor. And I don't have really time to get into this much, but the whole point of this figure is to show that at every single odor, the way that the animal is representing it is, say the green odor, actually in this case, the green odor is the one that the animal would be smelling at that time, but the representation that the animal has is showing the odor before it. And then as the trial goes on, it's also starting to represent the odor that goes on after it. But the only odor that the animal is smelling at that time is this green odor right here. And so you see that in this sequential learning regime, that the animal is representing the past and the future, as well as the present odor, which shows a nice, which at least gives me a lot of ideas of ways that this could be used. Particularly in reinforcement learning, there's a few ideas of how this could be used, which we can get into later. And I'm really excited about this work because I am just now about to finish a data set on a similar thing, but where these representations are coming online. So how does a novel, when I add novel information, so say like an odor sequence task 2, how does that change this representation? And in lieu of how is the new representation being altered? Lastly, this is some of the last stuff that I'm working on with is, as most people know, replay is a really common.
Ilan, we are running a bit out of time, so if you can be very quick on this.
Yeah, this should take 2 minutes.
Cool.
So replay is a lot that I've been working on right now. Most of us know that this is a common method used in continual learning to overcome some deficits to keep the network trained. And this has also been commonly used in rodent neuroscience. And so with the McNaughton Lab, we've been looking at sleep replay, as well as a bunch of other things. So this is a beautiful data set that we recorded from Cortex. And you can see that right here, the animal is awake, and here the animal is asleep, and you start to get this rhythmic sleep-like activity. And I'm also looking at the combination of the hippocampus with the rest of the cortex, particularly in this case is prefrontal cortex and hippocampus, and how during sleep information can be transferred between the two, which we can talk about later. So thank you to all the people that I work with, and thank you guys for listening, and looking forward to the rest of everyone talking.
Thank you very much, Kaylan.
Yeah.
So do you guys listening have some questions for Kaylan, or do we want to go to the next speaker and then have a debate or something at the end of the 4 talks?
We can wait to the end just so we know we can get through all the talks.
Okay, if anyone has a specific question now, you can ask it now if you don't want to lose the thread, or we can wait later. Cool. So I would say let's move to the next speaker. I guess, Edi Subutai, do you want to talk now?
Hello, can you hear me?
Yes, well.
Okay, great. Okay. Let me do this. Okay, can you see my screen?
Yes.
Okay, perfect. Okay, can you see it now? It should be full screen at this point.
Yes. Yes.
Okay, perfect. Thank you. Yeah, thanks, Vincenzo, for having me here, and it's a great session that you've organized. I hope everyone is staying safe these days. It is a strange time. Yeah. But yeah, so this my focus is it's really nice to have Keelan sort of give an overview of the hippocampal areas. My focus is going to be on the neocortex, which is the other big area of the brain that's pretty involved in intelligence and learning and memory and so on. And my focus is going to be on one specific aspect, which is sparsity. And what I'm going to do is kind of go in 2 different parts of my talk. The first part, I'm going to give a quick overview of certain aspects of sparsity that I think are specifically interesting for continuous learning. And generally speaking, sparsity is very deeply ingrained everywhere in the neocortex. In the second part of the talk, I'll talk about why or how sparsity can be important for continuous learning. You know, what can we learn in terms of learning rules, and how can we implement them in actual systems? And I'll talk about some results that show that sparse representations are very robust to interference and noise. I'll talk about some streaming data applications that we've done. And then we've just started looking at continuous learning in the context of deep learning for some speech and vision applications. So I'll just sort of give kind of the current status of that, although this is still very early in our in the cycle. Okay, so let's get going. I don't know if you can, if this comes out properly over the internet, but what you should be seeing is a video with lots of sort of flashing lights going on and off. And what this is it's a recording from the neocortex. I think this is in barrel cortex of a mouse. And the mouse is awake and behaving and engaged in tasks. And every time you see one of these flashing lights, there's a neuron that's actually spiking or sending out a signal. And the thing to notice here, so if you look at this kind of square area, it's jam-packed with neurons. And so the thing to notice here is that only a small percentage of the neurons are actually firing at any point in time. And indeed, your neocortex is just like this. You know, as you're listening to my talk, when you do speech, when you're engaged in any intelligent behavior or mathematics or language or anything like that, every part of your neocortex is extremely sparsely active, just like this movie shows. So what exactly is sparsity? What do we mean by sparsity? And generally, it means that stuff is mostly missing. So if you think about a sparse vector or a sparse matrix, it's a quantity where most of the elements are 0. And in neuroscience, most papers talk about sparsity in 3 different ways. So the first thing is population sparsity, where you can think about how many neurons are actually active right at this point in time. And that corresponds to the movie I was showing you. And the best estimates are somewhere around 0.5% to 2% of cells are active at a time. And if you think about deep learning systems, this is much, much sparser than any deep learning system. Lifetime sparsity is sort of related. It's sort of looking at a given neuron, how often does that cell fire? And if everything is sort of equal, then the population sparsity and lifetime sparsity should be kind of roughly equal. The third thing is actually connection sparsity. So if you look at layers of neurons or layers of cells, if you think about a layer kind of projecting to another layer, what percentage of the cells are actually physically connected? And it turns out this is also extremely sparse in the neocortex. So the best estimates is somewhere around 1 to 5% of possible neuron-to-neuron connections exist. And this is between layers that are directly projecting to one another. So this is also much sparser than most deep learning systems, where you typically have 100% connectivity between layers. But the connection sparsity is actually also interesting in that it's extremely dynamic. And not only is it sparse, but it's extremely dynamic. So it's changing all the time. And many neuroscientists think that a large part of the learning in the neocortex actually involves not necessarily adjusting the weights of synapses, but actually growing and removing synapses. So this is called structural plasticity. And here the network structure is actually dynamically altered during learning. And this occurs even in adults. So this picture on the left shows a section of a dendrite where connections are made, and each of these kind of bumpy things are synapses or connections. And this is the same patch of dendrite, same section of dendrite at 4 days apart. And wherever there's a blue triangle, that's where synapses kind of persisted from between these 4 days. And wherever there's a red triangle, that's where either there were new synapses or synapses were dropped. And this comes from a paper in 2015. And one of the most striking things here is that what they noted is every few days, 30% of the synapses were turned over. That is, there were 30% of the synapses were new, and 30% of the synapses that were there previously were no longer there. And this is an amazing amount of plasticity and network structure change, if you think about it. And what we think is going on is that essentially the system is constantly trying to learn and constantly trying to find patterns in the data stream that it's exposed to. And of course, most of those are going to be noise and dropped off, but some are going to be persistent and stay on. So this is a critical aspect of continuous learning in the neocortex. There's actually a lot more to sparsity than just these 3 things. So let's look at a couple of more aspects of it. One of the things to consider is actually the neuron itself. In machine learning, we think of neurons as being a simple linear weighted sum of inputs and an activation function. In biology, it's much more complex than that. What I'm showing here is a pyramidal neuron. This is the most typical, most common kind of excitatory neuron in the neocortex and in the hippocampus. And you can see it's got a pretty complex structure. Each of those kind of branchy things are dendrites. That's where the connections are actually made. And there are a couple of interesting things I want to point out. First is that if you look at the pyramidal neuron, it's actually a very sparse computing device. So if you think about these dendritic segments where connections are made, there are actually dozens of independent computational segments, and each of these segments can become active with just 8 to 20 active synapses. And if you think about the fact that thousands of neurons are projecting into this one neuron, the fact that 8 to 20 synapses can cause something to happen is actually, it's a remarkably sparse computation that's going on. And each of these pattern detections, if you will, are happening in parallel throughout the cell. This kind of figure shows an example of this, where if you activate 7 of the synapses, you get a sort of small response, but if you activate 8 of them, you get suddenly a nonlinear response. You get a high response. And interestingly, these are called dendritic spikes. These are responses that are internal to the neuron. They don't always lead to, in fact, they most often don't lead to an actual action potential or signal. The second thing that's related to it is that the learning in the neuron is actually extremely sparse as well. So each of these dendritic segments learn essentially independently. And when there's some sort of activity that happens, the learning is localized to the branch, to the dendritic branch that caused that dendritic spike. So if you look at the literature, you can kind of collapse it down into three very simple kind of learning rules. And it deals with whether the neuron actually spikes or not, or does not spike, and whether there was a particular segment that was activated or not. And the thing to remember here, these are very simple learning rules. They're continuously active. And in many cases, this is actually learning, again, involves growing and adding connections. So this, you can think of this as sort of learning rules that are sort of very fundamental to continuous learning in the neocortex. So this is a very quick overview of some of the aspects of sparsity here. This is part of a much longer talk. The thing to sort of, if you step back and think about it, you know, the population, the activity is sparse, the connectivity is sparse, neurons detect these sparse patterns within the neuron, the learning itself is sparse, synapses are very quantized, so the weights are sparse, and the energy usage is also correspondingly sparse because of all of these things above. And if you step back and think about it, this is nothing like deep learning systems. And you almost have to ask, you know, why are deep learning systems so dense? And the other side of this is, are there advantages to highly sparse systems that we can exploit? And at Numenta, we've been kind of working on this for a while. We think there's a whole list of things that that are critical to building intelligent systems that are kind of realized through sparsity, including computational efficiency and a bunch of other things. The thing I'm going to focus on, of course, is continuous learning here. So in the second part of this talk, I'm just going to try to take aspects of what I've talked about already and try to understand, okay, is sparsity important for continuous learning? And how can we build systems that are continuous learning that sort of exploit these properties? So the first thing I want to point out is that sparse high-dimensional vectors are actually extremely robust. So let's think about two sparse vectors, xi and xj. Let's look at the dot product followed by some sort of a threshold. Okay, this is kind of the most kind of primitive operations that's done in artificial neural networks. And if you take two sparse vectors, you can consider whether they match or not. And if their dot product is within the threshold, then we say it matches. And so think about sort of the gray circle here as being the universe of all possible sparse patterns. And the white circles represent all of the vectors that actually match each of those vectors. So you have x1, x2, x3, and x all the way up to xm, each with a sort of a circle of matching vectors around it. Now, as you lower the threshold, you're going to allow more robust matching in the sense that you can perturb a vector and the match will still succeed. But of course, the problem is now you're going to start intersecting with the matches of some of these other vectors that are out there. And the interesting thing is that as you decrease the dimensionality, as you increase the dimensionality of sparse vectors, what happens is that the size of the gray sphere, the set of all possible patterns, increases much, much faster than the size, than the rate at which these white circles increase. And what that means is that if you look at the ratio of the area of the volume of the white circles to the volume of the gray circle, that decreases exponentially. So these sparse vectors are actually incredibly far apart, exponentially far apart. So we can quantify this in an equation that's described in the paper. I'm not going to go through it in detail, but what this graph shows is kind of the probability that two random vectors are actually going to match or interfere with one another as you increase the dimensionality. And the y-axis is a log plot, the probability of a match, and you can see that essentially the probabilities decrease exponentially as you increase the dimensionality. So there's tremendous room in these sparse vectors, in the space of sparse vectors here. You don't get this if you match a dense vector against a sparse vector, for example, which is the flat dotted line up at the top. So this is exactly what you need to avoid catastrophic forgetting. You don't want new stuff to interfere with the old stuff that you've learned. And we think this is one of the fundamental aspects why humans can learn without forgetting older stuff. So we've tried to implement this in a couple of different ways. In the context of streaming data, we've implemented a system that has three basic components. I'm not going to go into these in detail. They're in the paper here. The first component is we created a model neuron that mimics these independent dendritic segments that are there in pyramidal neurons. We put these into a recurrent predictive network. So each neuron receives sparse input from the previous time step and is trying to predict the input at the next time step through its recurrent input. And then the third thing is these unsupervised continuous learning rules. And these exactly match the evidence from the neuroscience that I presented earlier. These are very, very simple local Hebbian style rules that you can implement. So we've tried this on a bunch of different streaming data sources. I'll show one example from a paper in 2016. This is a taxi demand data stream. So you can see that the taxi demand goes up and down on a daily basis, and the task is to predict the demand in the future, in the next few hours or the next day or whatever. And if you look at a bunch of different learning systems, the best ones are LSTM at predicting the next thing. So this is a batch train system. And the HTM, which is the network that we built, stands for hierarchical temporal memory. Its overall error rate is competitive with LSTMs as well. But what's really interesting is that the HTM network, because it's using sparse vectors and those continuous learning algorithms, actually adapt much more quickly to changing statistics. So the vertical dotted line shows the point at which the statistics of the network, of the data stream changed. And you can see that the LSTM and the HTM, the error spikes right after the change. But the HTM actually responds much more quickly and goes back to baseline error. Whereas for the LSTM, it takes a long time before the error goes back to baseline system. So this is an example of continuous learning in the context of streaming data sources. The second thing we looked at, and this is just very early at this point, is can we apply sparsity to deep learning systems? So if you think about a deep learning system, you have a linear weighted sum of inputs followed by some sort of activation function like ReLU. We converted this into a differentiable sparse layer. So we, instead of a ReLU, we do a k winner take all operation where the outputs of the top k units are maintained and the rest are set to 0. And the second piece is that the connection matrix is sparse as well. So most of the weights in the networks I'll show, or the network I will show, is sparse. And there are a couple of other tricks to getting this to work, which are kind of described in our 2019 paper down below. So we first tested this network on robustness and just general interference. This is an example from the Google speech commands dataset, which is released by Google a couple of years ago. It contains 65,000 one-word sort of commands spoken by individuals. And we've tried both dense networks and sparse networks on these. These are convolutional networks. The thing to see here is that the overall test score on the benchmark is competitive with dense and the sparse system. It's about 97% accuracy on the test score. The really interesting thing is if you look at the noise score, what happens if you add in random noise? The sparse networks do significantly better than the dense networks in handling noise, even though it's got many, many fewer parameters. And that goes back to the equations and the charts that I showed earlier. So sparse networks are inherently more robust to this. And we've done this with many other data sets in a lot of other contexts as well. And the last thing I'll show is we've just started thinking about, okay, can we take these sparse networks and build deep learning systems that try to avoid the catastrophic forgetting problem? And we've got some initial results here. We look at just the overall interference or the dot product between different input samples dependent on the layer of the networks. So you can see that along the x-axis, we're showing different layers. So we have the first convolutional layer, the activation function after the first convolutional layer, and so on. Along the y-axis, I'm showing the normalized dot product between vectors in the Google speech commands data set here. And you can see that right after the first activation function, the dot products drop. But for the sparse CNN, the dot products are at least in 1 or 2 orders of magnitude lower than the dense networks. And that again shows that with sparse networks, these vectors are much less likely to kind of interfere with one another. We also looked at the impact of dimensionality here. So this shows for a sparse network what happens as you increase the dimensionality. And you can see that if each layer has either 64 convolutional filters are 128 or 256. As you increase the dimensionality, the dot product drops by at least another order of magnitude, if not 2 orders of magnitude there on the right. So this is really encouraging. This shows that you can actually build deep networks with some of these sparsity properties, and perhaps combined with some of the other tricks that are known in the continuous learning literature, we might actually be able to build systems that can overcome the catastrophic forgetting problem. So that's pretty much my talk, and kind of summarize what emphasize the sparsity is deeply ingrained everywhere in the neocortex. Both activity and connectivity is sparse. Connections are continuously created and dropped, and that's a core piece of the learning that goes on in the neocortex. There are simplistic, sparse, continuous learning rules that are embedded within the neurons themselves that we might be able to actually translate to machine learning systems. And the second piece is that we think that for continuous learning, sparsity is actually critical. Sparse representations are highly robust to interference and noise. I showed in the context of streaming data that sparse networks can react very rapidly to changes in the statistics of the data set, much more rapidly than batch systems or dense systems. And we've started looking at deep learning in the context of speech and vision, which I didn't show today. And this is very much in process, but the initial results are kind of encouraging that sparsity could actually play a pretty big role there. Thank you.
Thank you so much, Subudai, for your great talk. Guys, if you have any questions specific to some topics of the last talk, you can say your question now, or we can wait and have the other 2 talks very quickly, and then have a debate and open question session. Okay, so I guess we can move on. I think that Eric has a very related topic, and he will talk about sparsity and continual learning as well. I am right, Eric?
Yep, indeed. There's some overlap. I think we have some new stuff to add as well.
Okay, so maybe you can go next so that we can.
Sure.
Okay.
Hold on, just need to figure out trying to do a window.
Meanwhile, if you want to discuss about these topics we are talking about today, you can join the work space, Slack workspace, where we have a channel for the meetups. It's called CL_meetups, or you can join the brand new discourse forum, in which we have a topic about this meetup, and you can discuss there your ideas, and give your comments, and look for the slides and the additional links I ask the speakers to put there.
Okay, unfortunately, the presentation doesn't show up in the selection menu, so I'll just do the entire screen after all.
Okay. I can see both. Share your entire screen or just a window. So I don't know how the other guys did it, but I think you can select a specific window and then the window.
Yeah, it gives me a bunch of other windows, but not the one that actually has the presentation in it. Like, it gives me my VS Code instance and stuff, but okay, this is a bit of a problem. I don't know if you can see anything yet, but
no, not yet.
Okay, this. See if I can get it to appear. I think it might be a Linux thing, to be honest.
Maybe we can move the Jeremy talk first, and then
sure, I'll figure this out in the meantime.
Yeah, exactly. I think you can share your screen as well, even though you're not presenting. And Jeremy,
yeah, I can go. Let me try. sharing my screen. Okay. Can you see?
Yes.
Okay, so I'm going to take a little bit of a different approach than the previous speakers, and I'm going to try to talk about more of a higher view of the building blocks that we know have an impact or a role in learning in biological networks. So I won't be able to describe the last 70 years of research in the next 10 minutes, but what I'm going to try is to give you a set of notions and concepts that with the aim at expanding the view and give new perspectives and new ideas to look for to build in artificial neural networks. And remember that the brain is an ensemble of very complex and very dynamical processes, and what we are studying is we use imperfect tools, and we don't have any baseline of what we're supposed to look for. So all of this is important because the information we gather is incomplete, it's messy, there's a lot of noise, there's a lot of unknown factors, and we only get tiny pieces with each experiment that we do. So learning can be studied at many levels. We can look at learning in behavioral changes. We can look at brain regions and the different interactions. We can look at different neural population. We can look at neurons by themselves and both functional and structural modifications. And we can also look at proteins and molecular pathways, which are all important. The challenge that we have is to reconcile all of these levels of studies and come up with a coherent view that can integrate information from all of those multitude sources. So as you can see here, for example, and it's only a set of examples, but we can look at learning at the behavioral levels and using a lot of different protocols and apparatus, and we can also do that when in a lot of different species. All of those give us information that we have then to put into a coherent ensemble. At the behavioral level, for example, we can do training that can be what we call mass or spaced, and which will result in a different memory length. So, for example, here in each of those instances, there's the same number of training, but as we can see, and you can focus here at the end, 5 days after the training, the animal that had a spaced learning remembered the task, whereas the animal that has a massed learning did not remember the task. So the behavioral training has an importance in how long a task is going to be remembered. And most paradigm, like this one, cannot be one-shot learned. Some can, and here, for example, fear conditioning is one of those paradigms which is used a lot for that ability to one-shot condition animal. But the use of a specific task is depending on what you want to look at in the brain, and the choice of the task is crucial. The next level is brain structure and their interaction, and that's I'm only going to speak about one example, which is that memory don't necessarily reside in the same brain region forever, and that's been talked a bit about previously. Here in the model of the hippocampal neocortical interactions during memory consolidation, a memory is going to be first encoded in the hippocampus, and then after a while, days, weeks, even months, it's going to be remotely stored afterwards in the cortex. And that's when a new memory is created. When you update an old schema, an old memory, you already have cortical modules built, and then you are going to update those on the fly from the hippocampus, and the dissociation between hippocampus and cortex is going to be much quicker than previously. So that's only one example. There are others. Also, the brain has been said before by Subutai has a highly sparse activity, and when encoding memories, only very specific subpopulation of neurons will endure long-lasting physical and chemical changes, which will make them the constituent of memory engrams, which are the physical substrate in the brain of a specific memory. So what's interesting there is that we now have the tools in neuroscience to look at those specific engrams. So for example, if you take a mice and you condition it with that one, in a fair conditioning, which is the mice is able to be conditioned with a one-time association of a shock with a sound, for example. And you can use some tool to label the cells that are activated. You can then use light to just activate those neurons without the context it was in, and the mice is going to react in the same way it did in the context. And so we can have a tool to dissect the molecular, the ensembles of cells which are activated by a specific memory. And here in the second part of the figure here, we can do a similar thing where we induce basically a false memory. We label the cells that are activated in a specific environment. We take the mice in a second environment, and we shock it while activating the cells of the first environment artificially. And then the mice is going to express fear in the first environment, but it was never shocked in the first environment. And so those memory engrams will be encoded very sparsely in the brain, and then they will consolidate, as we can see here. And then during retrieval, what's also very interesting is that that memory becomes labile again. So you can encode the same memory, or you can change that memory, and then it's going to be consolidated again. Another interesting thing is that some specific neuronal regions have an even more complex set of neuronal population, with new neurons being constantly created de novo, which is called adult neurogenesis. And that occurs in the mice, at least, in two brain regions, the hippocampus and the olfactory bulb. And those neurons, they integrate into pre-existing circuits without destabilizing the existing knowledge. And so its role has been shown, for example, to be important in pattern discrimination. And if you take two very image images with neurogenesis, you're able to discriminate those two. Without adult neurogenesis, you were not able to do that. And if you have an increase of adult neurogenesis, then the discrimination is even easier. So that's another, let's say, building block of how the brain is able to learn different things over time. Another level of analysis is the neuron themselves, and both the functional and the structural modification. So as been mentioned before, the neurons are highly plastic, and they have a lot of structural plasticity. They are highly dynamic structures, notably at the level of the dendritic spines, which is not the only level they are plastic at. They have the ability to remodel the dendritic arbor quite significantly, but a lot of studies have focused on dendritic spines, and dendritic spines come in many forms, as you can see here, from very thin filopodia-like spines to very big mushroom-like spines. And these spines here are the ones usually that are less stable, and also they are the less likely to be activated, meaning that even if they are here, they're not necessarily functional. On the other hand, the mushrooms-like spines are the most resilient to change. They are the ones that have the most impactful synaptic strength, and they are almost always functional. The other thing is that spines are not necessarily unit element themselves. There's a lot of homozygosticity going on. So for example, if you have a long-term potentiation protocol that activates this spine here, this spine is going to enlarge and stabilize. On the other hand, if you have an LTD on that spine can decrease in size, meaning its strength is going to be less important, you can also have propagation of this decrease to the next spine that's right beside it. And also you can have those spines here that are going to disappear completely, rejoining what Subutai talked about earlier. Next, you can also have a lot of wider type of plasticity, meaning that if you stimulate this spine, all the spines here that have an arrow, they are going to react to that stimulation and stabilize, grow in strength. But the spine here that was not stimulated is going to shrink. So you have, at the level of those dendritic segments, homostasticity, meaning that the overall strength of that dendritic segment is going to be stable. And then you also can have a lot of spinogenesis, so new spines, de novo spines that are formed and going to stay or be gone again later on. The next thing is not structural, but functional modification. And for example, this is about Hebbian plasticity, long-term potentiation, long-term depression, which I'm not going to go very detailed into. It's one of the most known thing in artificial neural networks that is used. So basically, long-term depression comes in when the neurons received stimulation after it fires, and the reverse interpretation happens when the neurons fire before the other one. And so this is associated at the synapse level, for example, interpretation by an increase in the number of receptors that are in the spine head, and that's correlated with the spine size, for example. So the bigger the spine size, the more it has receptors and the more strength it's going to have, if I simplify a little bit. And this is only the emergent part of the iceberg, meaning that when LTP occurs, there's also a whole lot of proteins that are activated, which lead to a cascade of activation, as you can see here, and that's only one overview. There are many different proteins that are involved, but all of these proteins will activate one after the other, leading to the remodeling that we just talked about, the structural remodeling and the functional remodeling. And they have, all of those have a very specific dynamic that is very much intertwined. And then we can go also, as you can see here, to all the way to nucleus modification, which are gene expression and gene upregulation, downregulation, meaning that some genes are going to be expressed that were not expressed, and the reverse is also true, which could impact the strength of the connections between two neurons at more or less long-lasting time windows. And basically this is to show the same thing. You have a whole lot of protein, a whole lot of complexity, which is not something you can see in artificial neural networks for now, in how those mechanisms, those long-term potentiation depression last in time. So in fine, that's all of that. What we can remember is that there's even more factors that I didn't mention that were mentioned before, like replaying memory during sleep. But learning is one side of the coin that I talked about right here. Forgetting is another. There's a whole lot of literature on forgetting itself, which is also very important to remember, actually. And also, the brain is messy, and some of these factors are important, have an important impact on the ability of an organism to perform ongoing learning and to remember for a long period of time. But others of those factors, not so much. We just don't know which one are the most important ones. And it's hard to know beforehand which one are important, which one are not. Also, biology has a lot of redundancy, so knocking out, for example, specific proteins will maybe not have an impact directly that you can measure. So as I said before, and as a conclusion, like the challenge is to reconcile all of these levels and come up with a coherent view that can integrate all the information from the studies of all these different levels. And I think modeling is the way to go to help us refine those biological models. And also the reverse is also true. Biology can help a lot those models to become more, to perform much better. Thank you.
Thank you so much, Jeremy. This was an amazing overview. Thanks.
Yeah, thank you.
And guys, do you have some specific questions you want to ask to Jeremy? So I have one. So how big it is the neuroscience community that is actually involved in continual learning, trying to modeling directly continual learning, not just memory, forgetting, all these different stuffs. So how many people do you think are really working in the context of neuroscience and computational neuroscience on this topic?
On continual learning specifically, I would say not many for now. The thing is, continual learning for the brain, for a brain perspective, is maybe not difficult to narrow, but the brain is always learning continuously, meaning that if you perform a learning or memory experiment, you have a time factor that's always there. So
yeah,
if I perform an experiment where I'm going to look at neurons and how they evolve around after a couple of days, for example, if I want to follow spines on a dendrite, well, that's continuous learning, even if you don't call it as such. So
yeah,
the whole literature on learning and memory in neuroscience is extremely extensive. Forgetting is also a whole lot of literature that you can go through, which is relevant to building artificial neural networks. So because a lot of neuroscientists don't specifically say they're working on continual learning, I would not say that their work is not relevant for continual learning.
Yeah, maybe I can add something to what Jeremy just said. I think I agree with him completely. Continuous learning is kind of the norm in all of these experiments.
Yeah.
In fact,
Yeah.
the type of learning we do in deep learning, batch learning, where you repeatedly go over a data set and do gradient descent throughout is completely foreign to neuroscientists. It's actually very hard for them to even understand that setup. So
at first it can be, yeah.
Yeah, exactly. Whereas in deep learning, continuous learning is a big challenge. We don't even know how to do it yet. So it's kind of interesting. So hopefully there's some promise that these worlds will intersect much more and we'll have more crosstalk.
Yeah. So maybe my question was more about how many people in neuroscience are really understanding that there is an issue in machine learning about this topic, and they want to help specifically address this topic, trying to bring their knowledge, trying to summarize, as you, Jeremy, and Subutai and Kaylan did, trying to summarize what's important to address this problem, especially from a biological perspective.
I'm not sure. I would not say it's widespread. It's definitely an interest, but I'm not sure how many neuroscientists would focus only on trying to merge biological and artificial neural networks in continual learning.
Yeah.
I would say more and more. I would not be able to say for sure how many.
If I could chime in for a second on this. So the McNaughton lab that I work with, we just had a DARPA grant, which was on continual learning almost exactly. And part of the grant, there was like 15, 20 labs, just on different parts. And I mean, DARPA is a big organization. And I was actually really surprised that there were that many different labs and organizations that are working on different parts. And maybe they kind of tuned their resource towards, DARPA's call to action was a continual learning regime. And predominantly they are computational neuroscience. But I think this issue is becoming more and more relevant. And even if it's not called continual learning, as Jeremy talked about in his talk, we're talking about different subsets. So you could research schemas. Schemas is becoming bigger and bigger in neuroscience as we go more and more. And you could talk about hippocampal replay, which is a huge area of neuroscience now, whether it's awake replay and ripples or whether it's sleep replay. So even though it's not under this continual learning umbrella, I would say that there are a good deal of labs that are working on this issue. And like we said, most tasks that an animal would complete, even reinforcement learning, which is another huge area of neuroscience, that's definitely a continual learning type problem that the animal has to solve. And so it's not branded under that umbrella, but there is a fair amount of work in the literature that people are working.
Hey, Vincenzo, it'd be cool if you proposed a workshop in some of these computational neuroscience conferences like Cosine to,
Actually, that would be
what's known in capture some of what's known in neuroscience for the specific purpose of addressing continuous learning and machine learning.
Yeah, I mean, I know that in the past, maybe you joined the previous continual learning workshop at Cosine the last year. It was not organized by me, but someone did. So maybe they will propose that again this year or next year. Cool. So maybe Eric, do you want to speak now? Did you manage to?
Second time's a charm. I basically what I did now is I opened. It didn't let me share my screen while others were presenting, but I opened up 5 applications at once, just showing the same presentation. Hope one of them gets captured.
Okay, let's try.
Let's see. Apparently the PDF is captured, so let's do that. Do you see it?
Yes, right.
All right, now let's see if the present mode works. Well, it doesn't really matter. Just don't want to break it at this point. So, all right, so I'm going to talk about bio-inspired continual learning. This will overlap a lot with what Subutai has done. But I think I can add some additional insight into it from more deep learning perspective. Because I myself am not a neuroscientist. I actually come more from the machine learning crowd, but I'm very interested in neuroscience and look up many things. And the algorithms we develop at Ogma Corp are based on things like Subutai mentioned, Sparse and stuff like that. But I'm not going to actually talk about our tech, but rather the principles that we think are important for continual learning. So, continual learning, deep learning. I mean, this is basically just a summary of the problem itself. We believe it's dense representations that deep learning uses. On the other hand, online continual learning is when you would have a data stream that doesn't have replay, but deep learning just kind of replays in big batches, and basically if you go beyond its replay horizon, it starts forgetting, and that's where you have to actually start using continual learning methods. So actually, there's a question that I was thinking about was, what is the difference between online and continual learning? So online learning is when you get an observation and integrate it with your existing data without breaking anything. But while continual learning is when you just in general incorporate more information without disrupting. And I think the difference is not very large. It's rather that online learning is at a lower time scale. Basically it's one sample at a time. You just push it in there and try not to disrupt other things. While continual learning is more broad and general, and you can have replay for a part of it, and then basically you defer the problem of online learning to later on, and then you start doing pseudo rehearsal and stuff like that. But I think it's also important to solve online learning, which is basically solving the problem at its core without deferring the problem to later on. So the question is, what does biology do? Well, a lot of this Subutai went over, but it doesn't use dense representations because this require IID assumption replay of basically exact copies. I mean, there's ways around this, which is what many continual learning methods do. But if you just think of DQN or deep learning style reinforcement learning, they usually just have a fat replay buffer of every memory ever and just randomly sample from that in batches. Our brain is very sparse, so very few cells active, and the inhibitory neurons are there for a reason. And we say that they decorrelate the excitatory neurons, and I'll get to that in a bit later. So also the brain can't support dense representations because they require way too much energy. And there's actually a paper here that I found quite interesting that talks about, specifically in the neocortex, how important sparsity is. But in general, we think that the brain uses sparse coding. This is, I think originally people started suspecting this from the Gabor filters in V1 of the visual cortex. And also, obviously, the readings you get, they exhibit both lifetime and lifetime and population sparsity. I mean, like, spikes are inherently sparse through time. So, sparsity and continual learning. We think that sparse representations basically solve it at its core, like basically at the online learning level, by removing the interference in the representations. This is a problem you have with deep learning, is that basically every neuron knows a bunch of things about a bunch of inputs rather than being special purpose for a specific portion of the input. It's kind of difficult to explain. You kind of get a feel for it when you work with it a lot. Especially if you like go for it on the Wikipedia entry for catastrophic interference. But sparsity is basically a trade-off between fully dense and grandmother cell style lookup table stuff. Grandmother cells, for those not familiar, is basically the original theory where it's so sparse that you have just one active cell that, say, represents your grandmother. And we argue that the trade-off between fully sparse and fully dense is actually not like a linear thing. Basically, if you go in between, you get the best of both worlds. Now, we don't have like any mathematical evidence, only empirically from within what we've done, and anecdotally from what we have, basically all the different kinds of algorithms we've tried. But it seems to not be a linear trade-off. So sparse coding in particular is a method for finding the both the codebook and the sparse codes themselves. We use SDRs and sparse codes interchangeably. I know probably Subhatai might disagree with this, but I think they're basically the same except one's binary, generally. But coding can often occur iteratively, and we argue that it does in the brain, meaning there's like a explaining away portion to it. And the dictionaries themselves are updated with STDP, which is just like longer-term Hebbian, shorter-term Hebbian. Now there's biologically plausible and implausible variants, and I have a little list of some that I found particularly interesting of the biological ones that I'll get to in a second. But they achieve similar results, basically. Those are shown on the right, like the Gabor filters that you get. That's the original Olshausen and Field paper from 1997. And like I said, the sparsity enforces orthogonality in the parameters and basically like bins things into their own separate non-overlapping representations. So here's an example. Oops, I think I skipped a slide. Yeah. So the way in biology we think it does it is basically you have some excitatory cells and inhibitory cells which learn to correlate with them. And as a result, they send back to the excitatory cells decorrelation or negative inhibition. And we actually think that plain case sparsity is not enough because you don't get the explaining away effects with that as well. So here's some models that do the explaining away, such as SailNet, which is a sparse coding system with spiking neurons. It's reasonably realistic, but it does have a important difference to EINet, which is even more realistic, in that it doesn't actually model the inhibitory cells separately, but just has inhibitory synapses instead, which is they basically have the same result. It's just slightly different, and one's more realistic. But the principle of the matter, which is having the sparsity and the explaining away, is still there. And then there's another one that's basically does the same thing again, but in a more deep learning fashion. And here's actually an example from the EINet paper of learning the codes in this biologically plausible fashion, where basically you iterate the cells and the E and I layers asynchronously, and they inhibit each other and basically settle on a representation over time. That's the explaining away process. And then you can update the weights with Hebbian rules, and you get these sparse codes. Now the question is it possible to just use backprop with these sparse representations? I mean, Subutai proposed one method, but we still don't think it's really a good idea for other reasons. But there's actually another method that is a bit different from what Subutai mentioned, which is that you just compute sparse codes with regular sparse coding algorithms in like an unsupervised hierarchy, and then you just route a deep neural network through the active units. So it basically selects subnetworks of the deep neural network. What's nice about this is that you can perform sparse computation relatively easily, meaning you only update the non-zero entries. It allows you to make tiny subnetworks and still train them with deep learning in a, like, for say, a classifier or something if you want, or to predict, say, a value function. And that's it. I try to keep it short. And augmentios are actual technology if you're interested in it. It makes use of these concepts. And that's our website there.
Thank you so much, Eric.
All right, no problem.
That was right in time.
Yeah, try to keep it short.
Great. So guys, do you have any question for Eric and his presentation? Otherwise, we will move to a bit of discussion just before leaving. So I have one question here on the chat, and I'm saying this out loud also because I think that the chat here will not be exported when we stream and we record this video. So it was a question from Jory Shosao. I don't know if he's here at the moment, but he asked a question to Subutai. With the current technology and instrumentation, is there observable variation in sparsity between individual organisms within a population?
Yeah, that's a great question. I can speak to that. So there's actually, if you look at, so there's a couple of ways I could take the question. One is sort of among different population, amongst different animals, and there is some variation, of course, between animals, but by and large, I think the numbers are remarkably consistent as long as it's the same experimental paradigm. The other way to take the question is actually within an experimental paradigm, if you look at a population of neurons, is there variation in sparsity? And there's actually quite a bit of variation. And this is a pretty long and involved topic, but maybe I could show you one. Let's see if I can I show one quick slide on this?
Of course, of course.
Let's see if that, can you see this?
Yes.
Okay, so this is from a presentation, a longer version of this presentation that I gave at Stanford a few weeks ago. And this is experimental data. This is a collaboration with Spencer Smith at UNC. He's now actually at UCSB. And he collected data from a couple of different brain regions. And what he did is he showed movies, a given movie to mice over and over again. So he would repeat the movie about 30 times. And what you can see at the top here is that on the first trial, there's it's a sparse set of activity, and neurons fire a fair. It's there is sparsity there, but on the last trial, it's much sparser. And on the right, we kind of quantified this as you showed more and more repetitions of the same movie. Initially, the sparsity was at about 1%, and then over time, it dropped down to about 0.4%. So what's going on here is as the animal becomes more and more familiar with the sequence and the stream of data, it needs fewer and fewer neurons to represent this. And this behavior is actually completely predicted by the HTM system as well, where the same thing happens as well in our system. And you can think about it from a predictive coding kind of standpoint, is that as you build up a model of your stream, the number of the difference between your predictions and the actual are going to get smaller and smaller, and you can actually represent the data with many fewer active cells. And in the beginning, when you don't know what's going on, there's a lot more error, and there's a higher density of neurons are actually required to represent it. So this is just one example of a type of variation that's quite interesting. But in general, yes, there can be quite a bit of variation. But in all cases, it's going to be much, much sparser than deep learning systems.
Yeah, it's interesting that you find that finding, because in biological experiment, when you look at immediate early genes, which are the genes that are activated after plasticity, the same kind of decrease in the activation of those cells that express those immediate early genes, you can see the same kind of curve with decreasing number of cells activated by the repetition of the same stimulus.
Okay, that's interesting. Yeah.
Okay, do we have any other question about any of the previous talks, or maybe you want to share some ideas about neuroscience-inspired continual learning, your own, anything?
I mean, I can point to a result that we just published if anyone's interested. On the website, I think I can post it in the chat. We recently were able to play Pong, like Atari style, on like a Raspberry Pi due to the savings you get from sparsity. Because it requires less processing power. And to my knowledge, no one else has been able to do that so far.
Great. So you can post all your links and the slides you presented on the Slack channel, journal, or whatever.
Sure.
Or you can maybe it's better because people from outside the Slack workspace can see it. You can comment your links and resources on a topic we have on this course on the forum of the association. So I have another question for the speakers. So any of you can respond to this. So what do you think would be the easiest thing to integrate in our continual learning systems that you say, okay, in biology is super important to do continual learning in the brain, and what this feature is super important. I would like to integrate these first in our continual learning systems that we don't have today, apart from sparsity, I would say. That's what I pointed out was very important.
I can maybe speak a bit to this. One thing that I know, so my old advisor at Indiana actually did this a while ago, where I was talking about oscillations, so different timings of when neurons are active. They did this to overcome catastrophic forgetting. So they implemented theta oscillation in a neural network, and they could overcome catastrophic forgetting. And it makes sense because you're turning off some weights at certain times, so you force the representation over to the other parts of the network, and then you turn on the weights that you just turned off, so you force the representation of those weights, and so you can minimize overlap in that way. And they found success by doing these kind of oscillations. And to me, it's kind of analogous to what like elastic weight consolidation does, but on a more spatial type of view, where elastic weight consolidation is kind of space agnostic, but it looks at the representation exactly. But you're essentially just turning off which weights already represent that part of whatever stimulus that you're giving to the network, or whatever input you're giving to the network. So I think that would be one really easy way to possibly implement, because you're effectively just turning off weights, and that'd be analogous to like what a neuromodulator might do, where you're kind of globally shutting off the plasticity of weights or turning up the plasticity of weights. So that would be one thing.
Yeah, maybe I can kind of add to that. if you think about plasticity in neuroscience, there are many, many different timescales at which plasticity happens. So there's really short-term plasticity of learning. There's kind of longer-term plasticity, which is closer to what we talked about. I think most of the people talked about. And then there's these neuromodulators, as Kaelan just mentioned. And I think all of those things, each one has kind of different roles to play, and trying to balancing them all together, I think might be pretty interesting in an artificial continuously learning system as well.
So don't you see any important features that are missing in terms of memory replay? Because we have talked a lot about sparsity and plasticity, but I think that memory replay is not something that you point out to be super important for continual learning in the brain, and it seems to be very important, at least up to now, for artificial learning systems.
I think I can talk to this a little. I think it depends on what time scale you're targeting, basically. Like I went over in my presentation, if you want to solve forgetting at like the lowest time scale possible, you need to use sparsity. But you actually, if in the end, if we ever build AGI or whatever, we probably need both, in my opinion, because the sparsity works well at the decorrelating individual samples, but even then, after enough time, you will still forget. It just reduces it a lot. And then you need some sort of replay, hopefully not just sampling a big memory buffer like we often do now, but like some pseudo-rehearsal, smarter methods like that, generative, adversarial network style stuff like that.
Yeah, I'll agree with Eric on this. I had to rush through my later slides, but that's basically what kind of one of the main pushes of our research is kind of looking into is memory replay. I mean, that was McNaughton's big paper, the McNaughton-McClellan paper, and so that's kind of an ongoing theme in the lab. And replay is definitely probably, I mean, if you think about what function it serves, right? The hippocampus is limited. So if you think of the hippocampus as the immediate storage location of immediate information, that's your short-term memory or like medium to short-term memory buffer, but cortex is your long-term memory repository, which you have a lot more cortex than you do hippocampus. You would need to push those representations off of the hippocampus so you can add new representations. So replay is probably the best mechanism that we have to do that. That's not to say that the cortex can't learn initial memories. We know that it can. We know that the cortex can learn some things, I mean, even without the hippocampus. That's been pretty well studied. But at the end of the day, replay does seem to be a central mechanism of how the brain at least solves the problem. So I don't think that's going to go away anytime soon.
Yeah, and I think one interesting aspect of that more and more data is coming out is kind of the importance of sleep and consolidation as a part of that whole replay experience. And so perhaps there's understanding from some of the tons of neuroscience experimentation that's been done on the importance of sleep-wake cycles and how we use replay to consolidate memories for kind of long-term retrieval that perhaps could play into artificial systems as well.
So thanks again to all the speakers. Jeremy, do you want to add something?
I was just going to add to that I would say it's difficult to know exactly which kind of building block to focus on because we don't really know the relative importance of each building blocks in the overall ability of artificial neural networks to perform continual learning. So it's possible one block have a much more important consequence on the ability to perform than another block, but it's also possible that it's the interaction of those blocks that makes the system perform much better. So, yeah.
Okay. So, does anyone has other questions to our speakers or to the rest of the Continual AI community?
I'll say one last thing just to kind of stir the pot. something that I've really been interested in more so in the last few months is, let's just assume that we solve catastrophic forgetting. What would be the next major hurdle in continual learning, for instance? Because you could, I mean, it's probably not going to be just, oh, we solve catastrophic forgetting, these neural networks can learn continually for the rest of their lives. So what are the next major problems that we might have to solve, and can we kind of foresee them and start thinking about them before we kind of have a nice mechanism for solving catastrophic forgetting? Is something that's really been kind of key on my mind lately.
Yeah, I can kind of give my answer to that, which is, I think you and others kind of touched on this earlier as well, and that is that dealing with streaming data and continuously time-varying data and sequences is an integral part of continuous learning, and that's not very commonly done in deep learning at all. So, and one of the ways we think that happens is through a prediction mechanism. That is, the brain is constantly making predictions about what's going to happen next. And when we actually see the inputs that come in, we can now compute an error. And through that error, we can kind of update our representations. And this allows you to learn in a completely unsupervised manner and hopefully very, very few labels. Today, I think deep learning is very, very dependent on these really large labeled batch data sets. And I think the natural next step of continuous learning is to think about kind of streaming data and doing unsupervised kind of predictive learning.
Yeah, absolutely. I agree with that. I mean, the next obvious step, for me at least, would be to move to non-ID data, so sequential data especially. So it's in line with what Subutai said. So it's, working with natural sequences. But I think one of the most important issue that we have now and we will have in the future after solving, let's say, catastrophic forgetting is trying to get to a level of generalization that is very difficult to build over time, because one thing is to remember things, one thing is to as you move on in time to being able to generalize your knowledge, removing what's just biased knowledge about the external world and trying to have the best performing model that you can build over time. I think this is not very straightforward. It's very difficult to do, even though you can still remember everything from the past. And that will be a major thing to work on in continual learning.
I have one more or two more things that might be interesting. Is as someone mentioned in the chat, short-term memory is another thing that you can also try to reduce the forgetting of. So that's a direction to go down, and also growing networks so that over time you start with few resources and then you continually add more resources. Spread, that's more in the far future though.
Yeah. So thank you all for joining for the second online meetup, Continual AI. You will find this recording of this meetup online on our YouTube channel. I will post the link on Slack and in our forum. So thank you again for joining, and please be safe during this outbreak that we have global. So thank you again. See you soon.
Thanks for organizing, Vincenzo, and stay safe.
Thanks, everybody.
Thank you so much for your talks.
Bye.
Bye.
Bye-bye.
Bye.